Thank you, Dr. Mohanty, for introducing me. Yes, it's my turn now. So I would like to reintroduce myself. I am Anukriti Srivastava from Banasthali Vidyapith, India. I'm a research scholar in biotechnology. So today's topic of discussion of mine is I'm going to find out the novel EST marker, that is expression sequence tag markers, associated with grain filling duration and tillering traits in wheat under terminal heat stress. As it is written that wheat is the world's third most cultivated crop, and it is consumed like really lot in India and several parts of different continents like Europe and several parts of people also consume wheat in Asia also, like they consume pasta wheat in the forms of noodles and different dishes. So China and India are the top two producer of wheat, and the origin of wheat was found in the region near the east in Holocene period. And I would like to tell that sowing time of wheat is mid-November to mid-December. That's spring wheat. We actually cultivate spring wheat there in India. So what's the problem with wheat is that there is a climate threat, like there is a lot climate threat of for wheat there in India. And of course, if we are talking about the climate threat, it is going all around the world. It's a worldwide issue now. Also, I would like to tell that heat stress reduces pollen viability and that causes sterility in plant. And there are 70 to 75% of carbs and there is protein 10 to 15%, followed by different vitamins and minerals. And wheat is highly sensitive to temperature above 30 degree, and we can see it manually. Like if we grow wheat in field under the natural condition, you will see there will be escape mechanism in the wheat, like it will roll its leaves or it will become waxy in nature. So you can manually see that okay, this is going to this is going the high temperature, and wheat is trying to avoid that temperature. Elevated temperature, especially during flowering and grain filling duration, can lead to reduced yields and grain quality in wheat crop. Grain filling duration is the important period for wheat because that ultimately decide that what is going to be your productivity or yield. And changes in rainfall patterns and increased frequency of drought can adversely affect the wheat crop. In Rajasthan, drought is also a major issue. Like period by period, it is decreasing. Like situation is still very improved. But if you see back in 15 years ago, you will see that there is a lot of issue of drought. But to escape that problem, we have started doing some climate smart program so that we can manage that situation. Also, now adopting improved agronomic practices such as crop rotation, conservation tillage, and optimized irrigation can help mitigate the adverse effect of climate change on wheat production. I would like to mention that there are a lot other gene editing programs that are also going on, like CRISPR-Cas9. I'm very much interested into that. Maybe I'll proceed with this technique in my future study. Because CRISPR-Cas9, that's what it do. It do what it will target some specific gene. For example, HSP gene that is responsible for checking the heat responsiveness in wheat or ultimately in any other crop. So what can I do? I will target HSP 90 or maybe HSP 70. I will target that gene and maybe I will improve or improvise something in that case. Or CRISPR-Cas9 is, I think I'm very fascinated with that technique. So for sure. Okay, again, I want to talk about what is the effect of terminal heat stress on wheat productivity. So As I told you that it is the third most cultivated crop globally. So what is the problem is like climate changes, particularly rising the temperatures. Uh, it poses the serious challenge for production. Also, it disrupts the double-rich stage, shoot root development, and early biomass. That is very dangerous because I wouldn't say dangerous, but also it will reduce your crop productivity because early biomass won't give you any weightage, won't give you good agricultural production, which is very important. Like we have to solve that hunger issue of the country also because the country is quite populous. So we have to change that situation also. And the study says that yield reduced by 6 to 51% under the high temperature. Also, rising temperature reduce photosynthesis, grain number, seed set, and crop yield. Also, heat stress affect depend on exposure duration and crop growth stage. It is very important point because we cannot say that, for example, if we sown wheat today, so we cannot say that okay, the heat stress starts from here. We have to see when it starts, like how prolonged wheat is under the natural stress. So it also depends like how much time are you given to that wheat to exposed with terminal heat stress. So here I'm talking about tillering and grain filling duration. These two are very important yield components. Plant tillers influence plant structure, nutrient efficiency, and biomass production, while grain filling duration affect the final grain weight and overall yield stability. Okay, so we are going to understand the genetic basis of these traits under heat stress, and it is very essential for developing climate resilient wheat varieties. Uh, what the tiller do is like tiller controls population structure. Again, I told you, and yield in wheat. Also, it stores organic matter, support root development. Organic matter, as such, I would like to say that starch accumulation also depends on that. Like your endosperm tissue development also depends on that. And okay, so stable grain weights maintained through compensatory effect between grain filling components with instability arising from variation in either. So grain filling components is very important because ultimately your grain weights will be stable or non-stable. It depends on that. Also, environmental fluctuations like temperature, water availability, light, and nutrition have caused significant variation in grain weight. That's so true. This is likely due to high environmental variability due to the long grain filling period. I tell you one story, like this is my third year of research and I did like two times, like two trials I did, but the result is not uniform. Like I'm not getting single uniform result. Every day the climate is changing. Even in every periods of minute. Like for example, if the temperature is 25 this time in India, it will be like maybe 41. In my hometown, I live in Prayagraj. It will be 41, and that's so drastic. And yeah, that's actually happening. That's the problem. Okay, so I was trying to show you some clips of grain filling duration and tillers, but unfortunately it is not supporting. So I would like to explain that this is actually what grain filling looked like when it started making grains, when it's developing spikes. It's not hard yet, it is soft because it is yet making the grains itself. And here I'm trying to, this is my hand actually. Here I'm trying to count the tillers, like what plant tiller looks like. Um, okay, so I would like to tell about the role of EST markers. Yeah. So I want to tell about the role of expression sequence tag. This is what the major work I have done. These markers are the molecular marker. They are derived from transcribed region of the genome. These markers are highly valuable in wheat breeding due to their association with functional genes and agronomically important traits. We have chosen EST markers because it is highly polymorphic. When it shows polymorphism, then only you can decide, okay, this is variation is going on. Also, this marker plays significant role in wheat breeding by facilitating the selection of superior genotypes on the basis of desired function. For example, if you want to conclude that okay, this wheat genotype is heat tolerant or this wheat genotype is like heat sensitive, you can always go with this marker. Also, research on EST markers associated with tillering and grain filling duration in wheat is relatively limited yet now. So it was my chance, like I got a good chance to develop something. Okay, so this is the setup. Okay, so this is the experimental design and methodology, and I will let you know that this all I have done with my own hands and in like too much sun. I did it with myself. I procured 16 bead genotypes from IIWBR, that is Indian Institute of Wheat and Barley Research, Karnal in India. Uh, these 16 wheat genotypes are from different agroclimatic zone of India. So what I did in crop season, generally spring wheat season, in December 2022 or 2000, till 2024, what I did, I sown 48 seeds in 3 replication. As you can see, this is how I did. Like I didn't do it like a farmer's, like random seed sowing. I poked the holes and just put one seeds in each hole. That was really tricky to do, but I did it somehow. And also I have maintained like some distance between them. I would like to say like some 5 centimeters to 10 centimeters difference, like equal distance, and that's what we call it precise phenotyping. Like it's not a random method, but a precise phenotyping. What I did is after that, after 50% of germination, extra plants were uprooted, and I have all like maintained 24 plants in each plot to avoid the border effect, so that like all plants get equal nutrition so that they grow easily. This work is done at Krishi Vigyan Kendra in Banasthali Vidyapith itself, Rajasthan. So this setup is timely sown and this setup is late sown. You can see the clear difference. Like this is the spring wheat, so it grows like really nice without heat stress. But this is growing like really small and really late. So this is like this is the difference. Next thing what I did is like I have designed some EST markers. Like I did some in silico work also. So these EST marker especially designed for terminal heat stress, and I would correlate with its different traits in wheat. But for now, I have correlate it with the tillers. What I did, first of all, I identified the candidate EST markers, which is actually related with heat stress, that actually responds with heat stress in wheat. These markers are selected based on their presence in genes involved in EHP like heat tolerance. Again, I said HSP 90 or 70. Also, we can also target like DREB gene. There is a DREB gene in wheat. But what I did, I actually went like go to the NCBI website. I copied wheat endosperm tissue sequences, and that's how I designed the marker. I'll tell you later. The next step would be like marker trait association. After designing markers, I try to correlate with this like various agronomic and physiological traits that I have recorded during the heat terminal stress. And then I tried for validation, because validation is of course very important with this SSR markers. And okay, so that's how I designed the in-silico EST markers. I use this software that is Primer 3 Web. It's a free software, and you can, I wouldn't say software, but it's a website actually. So you can just Google the website and copy your sequences. This is the sequence, copy your sequence, like desired sequence. I target wheat endosperm tissue because obviously there will be some heat responsive genes like HSP or DREP. So what I did, I pasted the sequence here, and then provide values of the parameter as per the requirement. So SSR markers need some specific requirement. Like if I'm going to randomly design marker, it will take a lot of time. So I have to make sure that I have to find that there should be a nice GC percentage, which is 60% above. That we usually target, like 60% above GC content is nice for having nice markers. And then what else is like, I have to find some different SSR motifs in that sequence also. I have to check the length also. I have to check the tandem repeats also. So these are the some requirements in the marker designing. And then you will click the pick primers, and this will design these markers, like these 4 markers. So you can see that I have some GC percentage here, like nothing is more than 60, but they are showing like 55 to 60 is a nice marker, like 55 percentage of GC and 60% like between is a good marker. So yeah, this is the web page. When you click the pick markers, you will get this web page and you will get these markers. So you can synthesize the markers, and then you will validate it like that. So first of all, what I did the DNA isolation. That's a basic method from CTAB. And then I did PCR amplification. So as you can see that this is the amplification they are showing. They have a different amplifications, like no, nothing is a same band, like it's a different bands, right? Okay, so this is the graph of initial and productive tillers, like in timely sown conditions. So as you can see, the p value is less than 0.05 because this is a nice value, like it shows that there is a strong significance between like my genotypes. Same here in late zone also. Here you can see, okay, this is the important part of my research here. What I did the marker scoring on the basis of 0, 1, 0, because this is how you do. And I have calculated HSI. HSI is the heat susceptibility index. So what I did, this is, see this band here, I consider that this is a 100 base pair because I put the ladder of 100 base pair. So this band I consider it is 100 or between 100 or 200. So it is the band here. So I consider this band as a 0, which is 0. And then you can see this HSI also shows 0. HSI value is important on the basis of 0 and 1, because when you say that 0 HSI, that means it's a tolerant genotype. So it is like it's totally correlated in that, like this is 0, the HSI is also 0, like less than 1. Here the band is above, so I mentioned it like 1, and the HSI stays 1.9, like it's more than that. So like this, like it is all like here it is absent because I found that there is a double band, so I'm not considering it. My study is only studied upon like single band. Also, I have found the phenotyping percentage, which is 52%. So if I found 52%, research says that is a nice marker. But I have yet find just one marker, which is correlating with this tiller. So yeah, this is the study. So the discussion and interpretation says that HSI helps to categorize wheat genotypes on the basis of response to heat stress. Wheat genotypes with a marker score 0 are classified as heat tolerant under terminal heat stress. As I said, like this is 0 and this is like below 1. So this is tolerant genotype, and same as all. Okay, the genotypes scoring more than 0 or 1 are categorized as heat sensitive due to reduced heat resilience. Marker scoring provides efficient method for distinguish heat tolerant and heat sensitive wheat genotype. This is a very basic method that you can predict that, okay, this is heat tolerant and heat sensitive. Also, this design marker can work as a putative molecular marker for terminal heat tolerance in wheat. I'm trying to find out more. I just have found one. And this marker-based approach enables like early selection of heat tolerant and wheat genotype for breeding programs as well. HSI-based classification help us to understand that wheat adaptability to terminal heat stress condition. Marker scoring methods offer fast and cost-effective also, and it is a reliable method also. And molecular markers provide accurate differentiation of wheat genotype based on their stress response. And this is the interpretation that EST markers for heat tolerance support future genetic research and breeding efforts for heat resilient wheat. So the conclusion is like in silico marker development for heat stress is like study developed in silico EST markers for identifying the heat responsive genetic regions in wheat. So actually this marker targets specific gene. It is not targeting the whole sequence or whole genome. That's why it is special. Also, there is a correlation with EST marker with wheat traits. The marker shows significant correlation with agronomic trait, like it shows significant correlation with the plant tillers. Also, it is the potential application for MAS, and also it enhance the wheat genetic resilience. Future research can be like validation across diverse wheat germplasm and environments in essential to confirm marker efficiency and strengthen breeding for climate resilience. So yeah, you can also develop climate smart wheat also. So yeah, these are the references, and this is my team. This is my supervisor. This is me happily roaming around in the field, and this is the poster presentation I did back in college. Yeah, thank you.